Okay, let's continue on the recording of chapter 2, animal physiology. Okay. So next is ligand receptor interaction. So now we enter the like a process of cellular communication. Okay, I have revised my map just now. So cell communicates. The modes is whether direct or indirect. So the concept for this direct or indirect communication, whether the chemical messenger involved or absent. Okay. So when we know chemical messenger involved in direct communication, we have to know the properties of the chemical messenger. So there are two properties, hydrophobic or hydrophilic. So hydrophobic chemical messenger and hydrophilic chemical messenger, these properties here determine the type of receptors um, use or binding. Okay, whether intracellular receptor or transmembrane receptor. Hydrophobic chemical messenger, the receptor is intracellular receptor. Hydrophilic chemical messenger, the receptor is transmembrane receptor. So this diagram here, to represent you the outside cell and inside cell. So the green color here, the chemical messenger. So whether the chemical messenger bind to the transmembrane receptor or intracellular receptor, depending on the properties of the chemical messenger. Now we will go into the process or response action of cell communication, that is ligand receptor interaction. So generally the interaction can be two, agonist or antagonist. Whether the interaction activate the receptors. Interaction meanings when the ligand or the chemical messenger bind to the receptor, it can whether activate or it block the receptor. So this diagram here just show you full agonist or partial. So full agonist ligand, it will cause large effect. But the partial agonist just small effect. So ligand here can also represent by chemical messenger or general name. Okay, so this diagram here. Okay, ligand binding. This binding, the ligand bind to the receptor, it will cause response. Okay, if non-ligand bind to the receptor, it will cause no response because it's not non-ligand cannot bind to the receptor because it's not compatible. Okay, it's the receptor, the ligand is not compatible to the this receptor here, so it will not bind to the receptor, so no response at all. So you can see here the B diagram here, agonist. So but a natural ligand bind to the receptor, it will cause response. And then agonist also, the ligand which is or which are an agonist, it bind to the receptor, will also cause response. Okay, it will activate the receptor. For antagonist, does not cause response at all. It can bind to the receptor but no response occur. Okay, so generally the interaction of the receptor binding can be natural, agonist or antagonist. Okay, so ligand bind to its receptor. So what happened next? Conformational change. This conformational change is actually the signal to the target cell. So let's go back to my map here. So the interaction of ligand binding to the receptor is actually the binding itself. Okay, let's put it in red color. So interaction of the binding of the ligand to the receptor. Okay, so interaction. Ligand receptor interaction. Next, we'll go to what happened after. This is the first one. Next, what happened after the binding is conformational change. Okay, happen to this. Okay. Okay. LR, ligand receptor. Ligand bind to the receptor, it will make the ligand receptor complex, it will cause response. More free ligand, Okay, so let's continue. Okay, ligand bind to the receptor, making this complex here, ligand receptor complex, it will cause response. So what happen actually? Okay, this is just to say if there is a free receptor and free ligand, it will keep binding and keep increasing the response. Okay, until the receptors become saturated, so no more receptors, so ligand cannot bind to any receptors, so the response will plateau here. Okay, so at this stage here, more receptors available for the ligand binding, so until reach plateau state here, because no receptor available for ligand binding. Okay. All right. Target cells vary in the number of receptors. So this slide here talk about the number of receptors or concentration of receptors. Target cell with high concentration of receptor is sensitive. Low concentration of receptor, less sensitive. Okay. The number of receptor here can change or can vary over time. Okay, it can vary. Yeah. Let's see what is the dynamic process here. It can become down regulation or up regulation. What is down regulation? Phenomenon to maintain homeostasis, where the number of receptors on the target cells decreases in attempt to reduce the intensity of the signal. So example here, the drugs abuser, okay, regular drugs user or drug abuser. Normal function of opiate receptor is to induce pleasure and to block pain. Okay, so when the drug abuser excessively consume drugs or excessively using drugs, the number of opiate receptor will reduce. Okay, the number of receptor will reduce. It will cause, will decrease. It will cause reduce response. Okay, reduce intensity of pleasure signal. Okay, um, if we can put like a value or magnitude here, let's say the intensity or the value of pleasure is 10. Okay, so 10 ligand bind to the 10 receptor. Okay, this is just like a silly example just to make you understand. Okay, this drug abuser consume drugs, so we can say maybe the ligand become 50, but the receptor still the receptor available is like a 30 or something. So excessive number of ligand binding to the receptor is abnormal for the body, so eventually the brain will instruct to reduce or to decrease the cell receptors. So from 50 receptor become less like 5 receptors. So excessive ligand only can bind to the 5 receptors. It lowers down or it reduce down the intensity of pleasure signal or the magnitude of the pleasure. Okay, so that is down regulate. The key concept is the number of receptors decreases, right? So next, the ligand receptor dynamics is up regulation. This one is vice versa to the previous one. Phenomenon to maintain homeostasis, where the number of receptors of the target cell increases in attempt to increase the intensity of the signal. Okay, increase the intensity of the signal, increases in numbers. Here we can say example, coffee lover. Okay, the coffee lover, adenosine here. Adenosine is the ligand, an inhibitory neurotransmitter functioning to reduce brain activity, producing calming effect. Yes. Adenosine is the ligand that binding to the receptor. This adenosine, if bind to the receptor, it will cause or it produce calming effect. We feel relaxed, sleepy, right? So this person here consume a coffee because caffeine in the coffee is antagonist to the receptor. So instead of adenosine bind to the receptor, caffeine bind to the receptor. Okay. So how the response? the brain respond to the removal of this calming effect, which is adenosine, cannot bind to the receptor due to caffeine bind to the receptor. So it will remove the calming effect by the binding also. In overtime, meaning overtime, the number of adenosine receptor will increase in attempt to intensify the signal. Okay, the normal situation is we feel sleepy at night, or yeah, because adenosine bind to the receptor. Let's say we put some values over there, like we have 50 receptors for adenosine. Okay, adenosine bind to the adenosine receptor. It will cause sleepiness or calming effect. Okay, but we have to do some work. We have to stay up, right? So we drink coffee. Caffeine will bind to the adenosine receptor. It will remove the calming effect, so we feel awake. Okay, let's say we have to stay awake. We have to stay up during the night at night, because we have to study maybe 2 weeks before exam, maybe. Okay, you the coffee drinker will notice that the first week, he or she only drink 1 cup of coffee, but the second week he or she have to drink 2 cups of coffee to get the same sense of awakeness. Okay, during stay up during at night. Okay, so what happened actually, the adenosine receptors number increasing during that 2 weeks, to increase the intensity of the signal. Okay, so it keep increasing. Why? To make adenosine able to bind to the adenosine receptors. Okay, so that's why coffee lovers need to drink uh, more cups of coffee to feel the same awakeness. Okay, I hope you understand this. So the dynamic of ligand receptor interaction, whether down regulate or up regulation, it depends on the number of receptor, whether decrease or increase. Okay, so signal transduction pathway. Okay, uh, but before that, we now talking about this here, the binding and then also the conformational change that cause signal, so interaction, right? Okay, after the binding of the ligand to the receptor or chemical messenger to the receptor, it will cause conformational change, and then it will cause signal transduction pathway. Why we call this one? It will convert signal or stimulus from one form to another. Okay, so this is the component of the what we call here, the receptors. Okay, we have the receiver part and transducer part. The receiver part, where the ligand binding to the receptor, ligand bind. Transducer part, where the conformational change. Okay, after this binding, it will change its conformation. And then amplifier is the signal transduction pathway, where it convert a substance to another form, and then it will convert to another form. Okay, and then responder, something that responds to the signal. Okay, this is just the diagram of the receptor. So we have the ligand binding, and then transmembrane domain or transducer, and sorry. So this is transmembrane receptor, ligand binding domain, transmembrane domain, and then intracellular domain. So, so you can see here, when the ligand bind, it will produce conformational change or occur or cause. Okay. All right. This is signal transduction pathway. Ligand binds the receptor, causing conformational change, or something happened to the receptor here, the shape change. Okay, because of the shape change, it will activate the substance A. Substance A, when it activate, it will activate substance B, and so on. Okay, so this is signal transduction pathway. Stimulus that convert one form to another, and then it will convert another form to another. Okay. Receptors undergoes conformational following to the binding of the ligand, change in the receptor shape here, and then activate substances. Okay. So signal transaction cascades can greatly amplify the signal. Okay. So that's the process or the action or the mechanism of what happened to cellular signaling or cellular communication. The binding, ligand receptor binding or interaction, it will cause conformational change. Eventually, uh, it will lead to number three, signal transduction pathway. So this is what happened in the cells, in all cells during their communication. Okay. So this later part here, types of receptor. So you can see we have ligand gated ion channel, receptor enzyme, G protein couple receptor. So here, these three here is transmembrane receptor. Why? Because it's stated or position or located at plasma membrane. So you can see here the last one here, intracellular receptor. So we have four types of receptors. First type, ligand gated ion channel. Second type, receptor enzyme. Third type, G protein couple receptor. Fourth, intracellular receptor. So this one intracellular receptor inside the cell. This one transmembrane receptor at the plasma membrane, located at the plasma membrane receptor. Okay, so the rest of this slide explaining each of these. Okay, I want you to read and digest this. We will have discussion during our session. Okay. Yeah. I stop here before I leave this part here, the later part of cell signaling, the type of receptor for you to study, to read. Okay. This one, this is facts. You have to read here. You have to read the books. Find this example here to clearly understand why is it intracellular receptor. Okay, and then ligand gated ion channel, transmembrane receptor. Okay, receptor enzymes. We have three types of receptor enzymes: guanylate cyclase, tyrosine kinase, serine threonine kinase. Okay. Okay, so this is information. Yeah, I leave this part here to you. Okay. Yeah. I expect question from you. Right. Thank you.